Good evening, everyone, and a hearty welcome to the fourth session of the lecture series Recent Trends in Biotechnology, organized by the Research and Postgraduate Department of Botany of St. Thomas College, Autonomous Thrissur, in association with the Department of Biotechnology, Government of India, sponsored by DBT Star College Scheme. So today, on this virtual platform, we all have gathered for the invited lecture by a very eminent resource person, Dr. Kiran A, on the topic Real Time PCR Applications and Significance. So, about our resource person, Dr. Kiran A, he's an assistant professor at the Center for Molecular Biology at Kerala Agriculture University, Thrissur, and he have completed his masters in agriculture biotechnology from Tamil Nadu Agriculture University and PhD in biotechnology from Rajiv Gandhi Center for Biotechnology, University of Kerala, Trivandrum, and his area of interest include plant pathology interaction, mainly transcriptome analysis, and he's famous for many national and international publications. So, once again, with immense happiness, welcoming the resource person of the day, Dr. Kiran A.G., for his invited lecture, Real Time PCR Applications and Significance. Hearty welcome, sir.
Thank you, Helena. Thank you, Helena, for the nice introduction. Today
Welcome.
we'll be discussing. Yeah, can you see the slides?
Yes, sir.
Yes. Today we'll be discussing real-time PCR, its application and significance. RT-PCR has become a household name after COVID pandemic. Uh, RT-PCR is actually reverse transcription PCR. For RT-PCR, for detecting coronavirus or any virus, even PCR is enough. But because of the higher sensitivity of real-time PCR, the detection, for detection of coronavirus, real-time PCR is used. Today I'll be describing the applications of real-time PCR and its significance. And in detecting coronavirus, it is one aspect of real-time PCR or PCR, and that is a diagnostic application of real-time PCR. I'll be touching on that, but I'll be discussing more about the research application of real-time PCR. In research, academic research, real-time PCR is mostly used for gene expression analysis. Gene expression analysis is very important because, as you know, although the genome or genomic information in all the tissues of organisms are the same, these different tissues differ because of the difference in gene expressions. So monitoring gene expression is very important for understanding biology and living organisms. Earlier there were many techniques for gene expression, as you might know, northern hybridization, then mRNA differential display analysis. All these were tools which were used for gene expression analysis. But now we have more advanced tools like transcriptome sequencing, microarray, and also real-time PCR. So these are the modern tools for studying gene expression. And in these modern tools, transcriptome sequencing and microarray, as you might know, they helps us to do a genome scale expression analysis. But real-time PCR, in real-time PCR, a genome scale expression analysis is not possible. And there we will be studying few genes. But if you look at the I mean, efficacy of the study, the most efficient analysis for studying gene expression, if we have few genes, if we have identified few genes, then it is real-time PCR. So real-time PCR is a technique which which is the most reliable technique for studying gene expression. When once we have zoomed in on a few number of genes which are relevant to our particular experiment. So today I'll be discussing in length on this application, that is application of real-time PCR for gene expression analysis. And also I'll be touching other applications of real-time PCR like uh, you know, detection, molecular diagnostics, and also allelic discrimination. So I'll share my slides. Is the slides visible?
Yes, sir.
Okay, so I'll start with PCR. All of you might know PCR. It is a revolutionary technology. Uh, PCR was invented by Mullis in 1985, and it was one of the revolutionary technologies in biotechnology, uh, and which actually led to all recent advancements like uh, whole genome sequencing and also CRISPR technology. All basically there are two technologies which revolutionize the way we do biotechnology. One is PCR, and another is recombinant DNA technology. Both these technologies, I mean, all the recent techniques, advanced techniques in uh, biotechnology, they are derived from these two technologies. So hence these techniques are very revolutionary in the way we do science, especially biology. And this I will not go into detail because you might know about PCR. So in PCR we use small oligos known as primers, which are specific to the region of the DNA that we want to amplify. So these primers or short oligos will be designed to the flanking region of the DNA that we have to amplify, as you can see in the figure. And then we do the uh, PCR, that is a polymerase chain reaction, wherein the in-between DNA gets amplified. And we get millions of copies of that particular DNA region, which is defined by these primers. So that's basically the technique. So what is revolutionary about this technology is that this technology helps us to clone a particular DNA fragment. A particular DNA fragment, we can make millions of copies of that particular DNA fragment, so that we can analyze that DNA. We can know what is the sequence and whether that contains genes or not. So that's the importance of this technique. In the same way, in recombinant DNA technology also, it helps us to amplify DNA fragments. So if you want to analyze the DNA fragments, that means if you want to know the sequence of that DNA fragment, we need to produce that DNA in millions of copies. So that's how these two technologies become revolutionary. And this also I will not discuss in detail. You might know this, the different steps involved in PCR. There are three different steps wherein the reagents are kept in three different temperatures. A high temperature, denaturation temperature for denaturing the template strands, then a lower temperature wherein the oligos will bind to their specific complementary sequence on the template DNA, and then those bound oligos will be extended uh, at a much higher temperature, around 60 to 74 degrees Celsius, which is the optimum temperature of the DNA polymerase, that is a tacked DNA polymerase. So these are the steps involved in PCR. And you might know this also, that is this tacked polymerase was isolated from Thermus aquaticus, which was obtained from hot springs. And only after the invention of this DNA polymerase, this entire process was able to I mean automate the entire process of PCR. Okay, now we'll discuss how the real-time PCR is different from the PCR, normal PCR. So in conventional PCR, what we do is we do a endpoint analysis. That means we do the PCR, and at the end of the PCR, we take out the PCR products and then analyze them by agrocell electrophoresis. So here we are analyzing the product only at the end of the PCR. So hence, in PCR, actually, there are different stages in PCR. You can see the figure. So initially, the PCR, that is the amplification, proceeds in an exponential phase. That means in each cycle, the product will be doubling. So initially, if there were 5 copies of your gene in the template DNA had 5 copies of your gene. In the next cycle of PCR, it will become 10, and in the next cycle, it becomes 100, like that. So exponentially, it will increase. I'm sorry, it won't double; it will exponentially increase. So the increase happens in exponential manner, but this exponential increase will happen only for a short duration of the PCR, say around 15 to 20 cycles. This exponential increase will stop, and then the PCR will move into a linear phase. This is because the reagents in the PCR mix gets depleted, and the depletion of the reagents, and most importantly the DNTPs and primers, that the depletion of those reagents also will happen in an exponential manner, because they are being used in an exponential manner. So as the PCR product increases in an exponential fashion, the reagents also will get depleted in an exponential fashion. So as the reagents gets depleted, the PCR reaction also slows down. That's why we see a linear phase here. And then if we still further keep the PCR for further cycles, we'll do the PCR for further cycles, the PCR will approach a plateau phase. And further, the PCR product itself will start degrading. If we keep after 40 cycles, the PCR product itself starts degrading. So that's how PCR proceeds. So this is how the amplification happens. So in normal PCR, we are analyzing the PCR product at the end of the PCR, that is after 30 to 40 cycles. So here we are analyzing the product at the plateau phase. So by this time, the reagents are getting depleted, and the reagents need not get depleted in the same fashion in all the PCR tubes, in all the samples that you are comparing. It need not get depleted in the same fashion. So that depletion of the reagent also will affect the PCR. So since here we are doing the comparison at the end of the PCR, here we will not be able to make any quantitative inference about the results obtained. What I mean is, so if we have done a PCR with different templates for a particular gene or a marker, so each of these is the PCR product from a particular sample. So here we know in this sample that gene is not present, and in these samples the gene is present. So in 2 of these samples, that is 3 and 5, the gene is not present, and in all other samples the gene is present. But here we will not be able to make a quantitative inference, like whether the gene copy number in the second sample is more than that in one, or whether this is higher than the gene number in 4. That kind of a quantitative inference cannot be made from PCR. So PCR is basically a qualitative analysis, where we can know whether a particular DNA is there in that particular sample or it is not there. And that DNA can be either a gene or it can be a marker. So PCR is endpoint analysis or qualitative analysis. But when even before real-time PCR were available, this PCR was used for doing gene expression analysis, and that is known as semi-quantitative PCR. So in semi-quantitative PCR, what is done is these PCR products are analyzed not at the end of the PCR. They are analyzed at an earlier cycle, that is in the exponential phase. So when the PCR is in the exponential phase, the samples are taken and then they are compared. So since the samples are compared in the exponential phase, there the reagents are not depleted. There will be sufficient DNTPs and primers in the reaction mix. So the reaction is being performed at optimum. So there, when we compare the products, we can make a quantitative inference about the expression of that gene in different samples. So this was the earlier method for gene expression analysis, wherein we use PCR, and it is known as semi-quantitative PCR. So this is a typical semi-quantitative PCR analysis, where you can see these are the different samples. These are the different samples, and the gene which we are whose expression we are studying is hexose transporter, And in this kind of analysis, we also keep a reference gene. That here the beta actin is taken as the reference gene. So reference gene is a gene which is not affected by your treatment. So these are the different treatments. These three are the different treatments, and uh, these are the semi-quantitative analysis for this particular transporter. So here you can see the PCR products are taken at earlier cycles. So these are the cycles. So PCR product is taken from 17 cycle, then 20 cycle. So after completing 23 cycles, the products are taken and then they are analyzed. So here you can see in 26 cycles, this sample has very less of the I mean, the PCR product is very less in this particular sample, but there is a band in this particular sample R0, and there is a significant difference is there. And when we compare the reference gene in the same cycle, that is a 26 cycle, the expression of actin is almost similar in both the samples. So here we can make an inference like this gene expression is getting increased in this R0 sample compared to the S0 sample. So so this is how semi-quantitative PCR is done. So this gives us this gives us an inference by quantitative analysis of the expression of that gene in these two different samples. And this is possible because we have taken the PCR product at the exponential phase. So in real-time PCR also we do the same thing. We compare the samples at the exponential phase instead of at the later cycles. So and also the importance of reference gene for any such kind of analysis, a reference gene is also required, and the gene that we take as the reference gene will have stable expression. It will not be affected by the treatment, or it will have the same expression in different samples. And why we use reference gene is that the reference gene is used in order to avoid the experimental errors that we create. That means the experiment involves different procedures like sample preparation, isolation of RNA, then quantification, then cDNA synthesis, and then finally real-time or PCR. So in these different steps, error can be introduced, human error can be introduced, pipetting errors will be there, and the enzyme efficiency may not be the same in different samples. All those can introduce errors. So in order to avoid those errors, we use a reference gene, and a reference gene should be a gene which is known to be stable with these treatments. So whatever these treatments may be, when these treatments are given, that is S0 or R0, when these treatments are given, that treatment should not affect the expression of reference gene. It should be stable. So this concept is important for real-time PCR also. Okay. Now we'll come to real-time PCR or Qpcr, quantitative PCR. So initially real-time PCR, real-time PCR basically involves real-time monitoring of PCR, hence the name. So here we monitor the increase in PCR product throughout the entire cycle. So so initially this for monitoring the increase in PCR product, ethidium bromide was used. Ethidium bromide, it is a DNA intercalating dye. We use ethidium bromide for visualizing DNA. So the same ethidium bromide was initially used for monitoring the increase in PCR product during PCR. And the first Qpcr machine was introduced by Applied Biosystem. You can see the machine. So in Qpcr, we have a real-time monitoring of the PCR. So this is how a real-time PCR result looks like. So here on x-axis, we have the cycles. Normally, real-time PCR is done for 40 cycles. So the data from 40 cycles is shown here. And on y-axis, you have the relative fluorescence units. So the fluorescence data is collected throughout the cycle. So this fluorescent data is collected at the extension step. So as I said earlier, PCR has 3 steps: denaturation, annealing, and extension. Just like that, real-time PCR also has these 3 steps, and the fluorescent data is collected during the extension step of each cycle. and then that is being plotted here. And here the samples are compared at the exponential phase. So I will explain that in detail later. So for detection, later more nuanced dyes were uh, identified. So for real-time PCR, there are two different chemistries which are used for detection. One is cybergreen, which is most widely used, and cybergreen is a DNA binding dye. It binds to DNA in a slightly different manner compared to ethylene bromide. Cybergreen binds to the minor groove of the DNA. Ethylene bromide intercalates between the base pairs. Cybergreen binds to the minor groove of the DNA. So it binds to any DNA. It binds to any double-stranded DNA. It mostly binds to double-stranded DNA, and it binds to the minor groove of DNA. And we have other methods also, which is using Stackman Props, and that I will discuss later in detail. So that's a different chemistry, and these are the two different chemistries which are used for in real-time PCR. Either it will be cybergreen or it can be Stackman chemistry. Stackman props are also known as hydrolysis props. We'll see why it's so. Okay, first we'll see the cybergreen dye. So as I said earlier, it is a dye which binds to the minor groove of DNA, and it has an excitation at blue region and emission at green region, and hence the name cybergreen. And it is more economical compared to the other. And this is the structure of cybergreen. The problem with cybergreen is that, as I said earlier, cybergreen binds to any DNA, any double-stranded DNA. So during PCR, we know the primers can anneal and form primer dimer. So primer dimer formation can happen during PCR. If the primer has complementarity at the 3' end, they can anneal, and then they can be extended by DNA polymerase, forming the primer dimer. So cybergreen will also bind to primer dimer because it is also a double-stranded DNA. And also, if non-specific amplifications are happening in your PCR, cybergreen will bind to those DNA fragments also. So if your primer is not specific, it can bind to other regions in the DNA in a non-specific manner. Then amplification will happen from those DNA regions also. So cybergreen will bind to all those DNA. So when we use cybergreen, the fluorescence will be coming from all the DNA, all the different DNA molecules in your sample. So when you use cybergreen, you have to be very careful. Your PCR is very specific. That means your primer is binding only to the intended targets, and there are no primer dimer. So that's very important when you go for cybergreen chemistry. Okay, next is Stackman props. Stackman props are expensive, but they are very specific. So these problems I discussed earlier of cybergreen dye, that's not there when we use Stackman props. So here in Stackman chemistry, in addition to the forward and reverse primer, we also design a probe, and that probe comes in between the forward and reverse primer. So here there is more specificity because we have an additional oligo which also binds to that particular gene or the DNA region which we have to amplify. There is more specificity, and this chemistry works by the mechanism, FRET mechanism, that is fluorescence resonance energy transfer. So this FRET mechanism, it is actually in this Stackman chemistry, the probe is another oligo. It's a slightly bigger oligo which also binds to that particular DNA region, and this probe is attached with a reporter dye and a quencher dye, and the emission fluorescence of the reporter dye will be the absorbance fluorescence of the quencher dye. So the reporter dye will absorb light at a particular wavelength, and it will emit light at a particular wavelength, and then the quencher will have the absorbance wavelength of the quencher will be same as the emission wavelength of the reporter dye. So the light emitted by the reporter dye will be absorbed by the quencher, and this will happen only when these two dyes are very close to each other. So when the reporter and quencher are attached to the probe, they are at close proximity, so this FRET mechanism can happen. That is, the light emitted by the reporter can be absorbed by the quencher. So, and this mechanism is known as FRET, fluorescence resonance energy transfer. And here in Stackman chemistry, the Tac polymerase that we use, uh, that Tac polymerase also has a endonuclease or exonuclease activity. It has a exonuclease activity. So normally in PCR, the Tac polymerase will extend the 3' end of primer, 4' as well as the reverse primer. Here in Stackman chemistry, the Tac DNA polymerase that we use also has a exonuclease activity. You might know exonuclease is an enzyme which can break phosphodiester bonds from the ends of DNA. So exonuclease can remove nucleotides from ends of DNA. So there are exonucleases which are specific to 5' and 5' DNA ends and also to 3' DNA ends, and there are non-specific exonucleases are also there. So here this Tac DNA polymerase also has a exonuclease activity. So hence what happens is the Tac DNA polymerase will extend the forward primer, and when it reaches the uh, reporter dye, when it reaches the when it reaches the probe, it will start degrading the probe. It will remove nucleotides from the 5' end of the probe. You can see in the picture here. So the Tac DNA polymerase will degrade the probe by its exonuclease action. So when this happens, the reporter and quencher will be separated. So the probe will be degraded by the enzyme, and the reporter and quencher will be separated. So no longer they are close together. So the fret cannot happen, and now the reporter fluorescence will be visible. That means the reporter will be fluorescing. That means it will be emitting light. It cannot be absorbed by the quencher because they are further apart. So as the PCR proceeds, more and more probes will be degraded. So, uh, so as the PCR proceeds, the fluorescence from the reported dye will keep on increasing. So in PCR, the PCR products will increase in an exponential fashion. So the reporter fluorescence also increases in an exponential fashion. So here, uh, the difference from Cybern chemistry is that here this is more specific because uh, this Stackman probe will be binding to the gene, to that specific gene. It is of more higher length than the primers, so it will be more specific. And so we are measuring the fluorescence only from the probe. So only when the probe binding happens, it will be degraded by the polymerase, the exonuclease activity of the polymerase, and the reporter will be freed and it can fluoresce. So as the PCR proceeds, more and more Stackman props will be degraded, and the reporter fluorescence also will increase. And if the PCR is increasing in an exponential fashion, the fluorescence also will increase in an exponential fashion. So here the fluorescence is coming only from the intended target, not from non-specific amplicons or not from primer dimers, because the Stackman props will not be binding to non-specific sites, non-intended sites, and also from primer dimer, Stackman props will not be binding to the primer dimers. So here the fluorescence is coming from only from the intended target. So Stackman chemistry is more specific, but it is very expensive compared to Cybern chemistry. So mostly in research labs, when there are a lot of genes, lot of genes are being studied, then mostly people go for Cybern chemistry. And once they identified some crucial genes, then for those genes, Stackman props are designed. So another advantage of using Stackman props is that we can do multiplex PCR. So what is multiplex PCR? So multiplex PCR is a PCR in which we can analyze many different genes in the same PCR reaction mix. So there we can use many different primers which bind to many different genes. We can do a PCR for many different genes together. because in Stackman Props, we can design Stackman Props with different reporter and quencher dyes. There are many dyes like wick, fam, etc. Many different fluorescent dyes are there. So we can design Stackman Props with many different dyes. And uh, in today's real-time PCR machines, it's possible to detect 5 different dyes. So multiplexing with 5 different dyes is possible. So 5 different genes can be analyzed in the same PCR reaction. So the advantage of this is that template, very little template is needed. That means with the same amount of template, we can analyze same quantity of template. We can analyze many different genes together instead of analyzing them separately. If we want to analyze them separately, we have to add template in 5 different tubes. Instead, here we are adding template in one tube and we are analyzing 5 different genes together. So that's one advantage. Then the sample to sample or reaction to reaction variation can be avoided. When we analyze genes in different tubes, the variations between tubes will be there. So that can be avoided when you analyze all the genes together in a single tube. But theoretically, multiplex PCR is good, but in practical terms, it's very difficult. Multiplexing 5 different genes is impossible, because five different genes means 5 different primers. 10 different primers will be there, and also 5 props will be there. So that much oligo in a single tube. That means there can be many dimer formation between these primers, and it will be very complex. Standardization will be very complex. So multiplexing is possible with 2 or 3 genes. Beyond that, it will be practically impossible. So that's a comparison between the two chemistries which are used for real-time PCR. Okay. Then now coming to Qpcr machine. In Qpcr machine, there is also a thermal block, just like normal PCR machines. There will be a thermal block. And in addition to that, there is an optical module in Qpcr machines. And this optical module consists of an excitation source or many excitation source, and an excitation filter, then an emission filter, an intensifier, and detector. These are the different components in the optical module. And multiplexing with 5 different fluorophores are possible in present-day real-time machines. Okay, now we'll come to data analysis. So in real-time PCR, as I said earlier, we'll be collecting the fluorescence data from the PCR reactions at the exponential phase. So in each cycle at the exponential phase, the fluorescence data will be collected from the PCR reactions. And the final result looks like this, wherein these fluorescent data will be plotted. So x-axis will have the different cycles, and y-axis will have the fluorescence values, fluorescence units. And here, the basic data that we use to compare different samples is known as CQ value. So as I said earlier, when we do gene expression analysis, it's very important, or if we do any analysis with PCR, quantitative analysis with PCR, it's important to compare the samples at the exponential phase. So in real-time PCR, we do just that. So in real-time PCR, we have the fluorescence data collected throughout the cycle. So we have the fluorescence data collected throughout the cycle, and then we'll fix a threshold fluorescence. This threshold fluorescence can be any value, any arbitrary value, but it should pass through the exponential phase of all the PCR reactions that we are comparing. So here there is only one PCR reaction shown here, and this threshold fluorescence should be set such that it will pass through the exponential phase of the PCR. So you might think why the PCR is increasing only after say 15 cycles and not at the initial stages. So actually the PCR, in PCR, the amplicons or PCR product will increase in an exponential manner from the first cycle itself. But in real-time PCR, the increase in fluorescence of cybergreen or Stackman chemistry will be visible only after some cycles. This is because the unbound dye also has a fluorescence. The unbound dye has a fluorescence, and that is known as basal fluorescence. And only when the fluorescence from the PCR product increases above the basal fluorescence, it can be detected. That's why we see an exponential increase only after some cycles. So here we have the curve, and we should place this threshold fluorescence for comparing different samples in the exponential phase. That's the most important part. Here you are seeing only one sample, and you can fix this threshold fluorescence anywhere in this graph, and that will give you the basic data of real-time analysis, that is CQ data. So what is CQ data? CQ data is the fractional cycle at which the amplification curve touches the threshold fluorescence that we have fixed. So that's the fractional cycle number where the fluorescence from the PCR reaction reaches this threshold fluorescence. So that is the basic data that we get from a real-time PCR analysis. So if the initial copy number of your gene is higher, if the initial copy number of that particular gene is higher in your sample, then your sample will take less number of cycles to reach that particular threshold fluorescence that you have fixed. And if in another sample the copy number of the gene is lesser, then that sample or that PCR reaction will take more cycles to reach the same threshold. So that's how real-time PCR helps us to compare the amount of gene in different samples. So once again, a sample which has less copies of your gene will be taking more cycles to reach that particular threshold fluorescence, and a sample with more of that particular gene, more initial copies of that particular gene, that will take less number of cycles, less number of PCR cycles to reach the same threshold. So if we get a higher CQ value for a particular sample, then that means that sample has a lower copy number of that particular gene. And if we get a lower CQ value for a sample, that means that sample has a higher copy of the gene. So that's the basis of comparative quantification of gene expression in different samples. So here I have explained the plot. So there is a baseline because of the basal fluorescence, and then the exponential increase of the PCR will be visible, and we have to set a threshold fluorescence. It can be any arbitrary value, and only thing that we have to take care is that threshold should pass through the exponential phase of the PCR. So there is no way to identify the exponential phase, but we know from the curve it's possible to see the linear phase starting. So you should not keep the threshold fluorescence at that linear part of the curve. It should be kept in the exponential phase, and if we keep your threshold very low, then what happens is the baseline estimation, it is not good to keep the threshold very low, because there also although the PCR is happening in an exponential manner, that will not be reflected in the curve. I will not be going into the detail. There is a baseline subtraction of this data, and this baseline subtracted curve is actually used for estimating the CQ values. So that process, that baseline estimation process, will also that process will affect the data points at the base of the curve. So the point that has to be considered is, when you keep the threshold should not be very low, near to the baseline. It should not come lower to the baseline. That data points need not be exponential, and it should not go very high to the linear phase of the PCR. So if you keep the threshold anywhere in that exponential region, uh, although the CQ values will change, but the relative CQ values of your samples will not change. The CQ values of individual samples will change, but their relative values will not change if we keep the threshold in the exponential phase of the PCR. So it is these CQ values that are used for analysis, any analysis, gene expression analysis or allelic discrimination. So this is the CQ values that are used. So this is the basic output from a real-time PCR. We'll discuss the different types of analysis later. Before that, using real-time machine, there is one more analysis, that is Mel Curve analysis, and this is done after the PCR. So just like after PCR, we check the PCR products in agarose gel by agarose gel electrophoresis. In real-time PCR also you can do that, but here there is one more analysis called Mel Curve analysis, which does the same, which helps us to identify whether there are non-specific amplifications or whether there is primer dimer present in your PCR product. So that can be checked by another analysis known as Mel Curve analysis. So this analysis helps ensure the specificity of the amplification. That is, there is only the intended PCR product. There are no primer dimers and non-specific amplifications. So this is performed after PCR. So what is done is the PCR products is subjected to different temperatures, starting from a lower temperature of 60 degrees Celsius and gradually to a higher temperature of 90 degrees Celsius. So after PCR, the PCR products are subjected to higher temperature starting from 60 to 90, and the temperature is increased in an incremental fashion, like from 60 degrees Celsius, the fluorescence is taken, and then temperature is increased to 60.5, again fluorescence is taken, then it is again taken to 61, like that. In a 0.5 degrees Celsius increment, the temperature is increased, and at each of these different temperatures, the fluorescent values are taken. The fluorescence from the PCR reaction mix is taken, and then that will look like this. So initially, this actually represents many different samples. Each line represents the fluorescence from individual PCR reactions. So initially, all these PCR reactions will have the PCR products. So if you are using Cybern Dye, the Cybern Dye will be bound to the PCR products and we'll be getting a fluorescence value. And as we increase the temperature, so in this graph, as we go from left to right, the temperature is being increased. So as we increase the temperature, the PCR products will start denaturing. And as the PCR products denature, the fluorescence also will decrease. So Cybern will be binding to double-stranded DNA. So as the PCR products are denatured, as they become single-stranded, the Cybern will not bind to them in a strong fashion. So the fluorescence also, the fluorescence of Cybern comes only when it is bound to DNA. So as the PCR products are denatured, they become single-stranded DNA. The Cybern will not be bound to those DNA. So the fluorescence from Cybern also keeps on decreasing. And when the melting temperature of your PCR product is reached, the PCR, half of the PCR product will be denatured, and that's when there is a drastic reduction in the fluorescence. And after this, there are only single-stranded DNA. The entire PCR product will be denatured. And by taking a derivative of these values with temperature, we will get a graph like this. So the dip here, derivative of that will give a peak like this. And this peak actually represents your PCR product. It represents the denaturation of your PCR product. So in this particular PCR, we are seeing only one peak. That means it is a specific amplification. You have only one PCR product. It's like you seeing a single band in agarose gel electrophoresis. So after PCR, you do a agarose gel electrophoresis, and if your PCR is specific, you'll be seeing a single band. So this is just like that. And if there was a primer dimer we will be seeing another peak here, a smaller peak of primer dimer here, because primer dimers are normally smaller than your PCR product. So it will denature at a lower temperature. So we'll be seeing a peak here. And if there are non-specific amplifications, then you will be seeing multiple peaks. So that's how Mel Curve analysis helps us to ensure the specificity of our PCR. So in real-time PCR, there is no need to do a agarose gel electrophoresis. You can do a Mel Curve analysis and see the specificity of your PCR. Okay, now we'll come to the different analysis using Qpcr. So there are mainly three different analysis using Qpcr. There is absolute quantification, relative quantification, and allelic discrimination. So Qpcr we use for these three different kinds of analysis. So the most widely used one for research purpose is relative quantification, where we are interested in comparing the expression of gene in different samples. So that's the that's the purpose for which we use PCR in research. But in diagnostic labs, you use Qpcr for detecting pathogens, etc. So in that case, it becomes if you need to quantify, then it becomes absolute quantification. And even relative quantification and absolute quantification can also be used for gene expression. So in absolute quantification, you are interested in identifying the actual quantity of the DNA, or how many copies of genes are there in your sample. That's when you go for absolute quantification. So it's just like a estimation analysis done in biochemistry for estimating any compound. It's just like that. So you'll make a standard curve with known quantities, and then from that standard curve you'll estimate the amount in the sample. So just like that, you can do for gene expression studies also. So absolute quantification, when we need to know the actual copy number of your gene. But normally, when we compare gene expression, we normally are interested in looking at the expression of the gene in a control sample, expression of gene in treated sample compared to that in control sample. So if we need to know only relative expression of a gene in control and treated sample, then we can go for relative quantification. So that's the most widely used method for gene expression analysis. And then for allelic discrimination, that is marker analysis, most importantly SNP marker analysis, and this allelic discrimination is used. So if you have two different alleles of SNPs, then you can design different Stackman props to amplify these two different alleles, and then you can do a allelic discrimination analysis in the population. That is for genotyping, most importantly SNP genotyping. So these are the three different analysis involving Qpcr. So first, I'll discuss absolute quantification. So as I discussed earlier, it is just like the estimation that we do in for biochemical analysis using a standard curve. So here we are interested in knowing the exact copy number of that particular gene in that sample. So here we will have known quantities of that particular gene, and using that we will create a standard curve. So we will have different PCR reactions with different quantities of that gene, and we will do a real-time PCR analysis. So you can see here the amplification curve for the standard curve for the different standards. So these are the amplification curve for the different standards. So using the CQ, so here also we take the CQ values, and using these CQ values we will create a standard curve, and then from the standard curve, and this blue one is the unknown. This is the unknown one, where the gene quantity we have to estimate. So using the CQ value of this unknown sample and the standard curve, we can estimate the copy number of that gene. So this is how absolute quantification is done using a standard curve. So so this is not a widely used method, and this can also be used for gene expression. So here we'll get the actual copy number of that gene in the different samples. And for forensic samples, for detection of samples, this absolute quantification is done if the quantification is also required. Quantification of that particular gene is required from the forensic sample, then absolute quantification is done. Then another important application of absolute quantification is determination of transgene copy number. When transgenic plans are produced, the transgene integration is a random event. If we go for agrobacterium-mediated transformation or bialistic method, the integration of the transgene is a random event. So multiple copies of the transgene can get incorporated in the plant. And we are not interested in such transgenic plants. So we are interested in only single copy integration. So the transgenic plan, which will be further advanced for developing varieties and all, that should have a single copy integration. So in order to ascertain that the transgene has a single copy, the transgenic plan has a single copy of transgene, the absolute quantification can be done. So here a standard curve is generated using plasmids carrying the transgene. So different copies of different non-copies of the transgene is taken and a standard curve is created. So the plasmids which carry the transgene can be used for that purpose, for preparing the standards with different copy numbers of the transgene. And then using that, we can do a standard curve analysis and determine whether the transgene has single copy or multiple copies of the transgene. So absolute quantification is used in this case. Then another application of real-time PCR is allelic discrimination. As I said earlier, mostly it is done for genotyping using SNP markers. So here we are interested in looking at genotyping the population for different SNPs. So SNPs, we know those are DNA regions with a single base difference. Like in this particular DNA loci, in some individuals of the population there is C, and in some other individuals there is T. So these are two different genotypes, and this is genotyped using, and these are known as SNPs, and it can be genotyped using real-time PCR or Qpcr. So here we cannot go for cyberine chemistry. Here we have to use Stackman chemistry because cyberine binds to all double-stranded DNA. So here for SNP allelic discrimination using SNPs, what we do is we design two different Stackman props, one which binds to one of the alleles of the SNP and the other which binds to the other allele of the SNP. And these two props will be attached with different fluorescent molecules. So here this probe is attached with Vic and this probe is attached with FARM. So these two have different fluorescence. And when we do a real-time PCR, we can differentiate which populations, which individuals of the population has a TT genotype and which has CC genotype and which are heterozygous, which has both these genotypes. So that can be differentiated. So this is how allelic discrimination assay looks like. So this is another application of real-time PCR, and this can be done only using Stackman props. And now we come to the third application of real-time PCR, that is relative quantification. So this is the most widely used method for gene expression analysis. In most of the gene expression analysis, we are interested in comparing the expression of the gene in treated compared to control. And such analysis is known as relative quantification. And the method used for relative quantification is known as comparative CQ method or delta-delta CQ method. So in delta-delta CQ method, we will be taking CQ values of the target gene, that is the gene whose expression we have to analyze, and we also take CQ from a reference gene. So already I have mentioned the importance of reference gene in the case of semi-quantitative PCR. So here also the purpose is the same, to cull out the errors that are introduced during the process, that is the entire process, RNA isolation, cDNA synthesis, etc. So in order to remove that errors introduced from the data, we use a reference gene. And the reference gene should be stable in response to the treatment. Whatever treatment that we are giving, this reference gene expression should not be affected by that particular treatment. That's very important. So here we will be generating CQ values for the target gene as well as the reference gene. And we will be generating CQ values from control sample as well as the treated samples. Okay. And then a normalized relative quantities are calculated, uh, and it is calculated by a delta CQ method. So normalized relative quantity is calculated by this equation, that is 2 raised to minus delta CQ. We'll see that in detail. So delta CQ stands for delta CQ treated minus delta CQ control. So before that, delta CQ is the CQ of target gene minus CQ of the reference gene, that is delta CQ. So in delta CQ method, we subtract. In delta CQ method, first delta CQ stands for CQ of target gene minus CQ of reference gene. So as I said earlier, we take CQ values for the target gene as well as CQ value for the reference gene. This will be collecting from both control and treated samples. So with control sample also we do a PCR with the target gene and also with the reference gene. With treated sample also we do a PCR with target gene as well as the reference gene. Now we calculate a delta CQ for the control sample and the treated sample. So the delta CQ is calculated like this. The CQ of the target gene, CQ of the reference gene is subtracted from the CQ of the target gene to calculate the delta CQ. So like this we calculate a delta CQ for the control sample as well as the treated sample. Now next delta CQ is delta CQ treated minus delta CQ control. So this delta CQ is the normalized value. Normalized means the normalization is done with the target gene values are normalized with reference gene values. That means all the errors that are introduced by the process, that is being removed by this normalization. So this delta CQ is the normalized value, normalized gene expression value in the treated, and delta CQ control is the value for the control. Now for calculating the relative value, that is the value in treated compared to control, we do delta CQ. So delta CQ involves delta CQ treated minus delta CQ control. So that will give you delta CQ, and delta CQ is the normalized relative quantity in treated compared to control. So this is how in relative quantification the relative gene expression in the treated is calculated compared to control. And here this equation is used for calculating normalized relative quantity, and the equation is 2 raised to minus delta CQ. So already I have explained how we calculate delta CQ, and this is only cycle difference. So this is the normalized cycle difference, and in each cycle of PCR the increase happens in an exponential fashion. So whatever the product in one cycle, in the next cycle of PCR that product will double. So in each cycle it will double. So in the first cycle if there are 5, in the next cycle it will become 10, in the next cycle it will become 20, like that it will double. So that is why this delta CQ is taken when power 2 to the power of delta CQ. That is the actual difference in the gene copy number, because delta CQ is just the cycle difference. So in each cycle, if the PCR is happening at a maximum efficiency, then the PCR products will double. So if there is a difference of 5 CQ between your samples, if there is a difference of 5 CQ between the treated and controlled samples, then the actual difference in the gene copy number will be 2 raised to 5. So That's how the equation becomes 2 raised to minus delta delta CQ. And also the minus here because CQ is inversely proportional to the copy number of that gene, actual copy number of initial copy number of that gene. As I said earlier, if the initial copy number is less, CQ will be higher because the PCR will take more cycles to reach the threshold fluorescence. And if the initial copy number is high, then the PCR will take less cycles to reach the same threshold. So here CQ has an inverse relationship with initial copy number of your gene. That's why here we give a minus. So that explains the equation, and this will give the normalized relative quantity in treated compared to control. And I will explain that with an example, then it will be more clear. Okay. So here in relative quantification, a minimal relative quantification looks like this. So there will be replicates will be there for each of this. Replicates will be there. So here I haven't shown the replicates. This is a minimal relative quantification assay. So there should be a control, a treated, and then a target gene, then a reference gene. And in each of this case, you can take replicates, and that they are known as technical replicates. And both the target gene and reference gene are amplified in control and treated, and we'll be generating CQ values. So these are the CQ values obtained, and then first we'll calculate the delta CQ, that is the normalized values in control and treated. So delta CQ is CQ of target gene minus CQ of reference gene. So in control it will be 28 minus 20, that is 8, and in treated it will be 24 minus 19, that is 5. So these are the normalized values. So your target gene values normalized with the reference gene. Now we will calculate the delta delta CQ, that is delta CQ treated minus delta CQ control. So this is the relative value in the treated compared to control. So that's delta CQ treated minus control, that is 5 minus 8. So it will be minus 3. So the value for treated delta CQ treated minus delta CQ control will be 5 minus 8, that is minus 3. So this is the difference in CQ value in treated compared to control. Now this is just the difference in CQ value. Now you have to find the difference in the expression of the gene. So for that we have to calculate 2 raised to minus delta delta CQ, and that is 2 raised to 3, that is 8. So your treated has 8-fold more expression of that gene compared to your control. So that's the final inference from the study. And in a histogram it will look like this. Your treated has 8-fold expression, 8-fold more expression for that particular gene compared to your control. So this is how relative quantification is done using real-time PCR, and this is the most widely used method for gene expression analysis. And earlier people used to do PCR and in I mean without any standards and in a proper manner. So because real-time PCR, you should understand, if you do real-time PCR, you will get some data. Whatever way you do, whether you do it correct or not, you will get some data. But whether that actually reflects the conditions, that depends on how perfect you have done your assay. That's very important, because you will definitely get a result, but whether that will reflect the actual actual conditions, that depends on how perfect you do the experiment. That's why in 2009 all the scientists who were involved in real-time analysis, they actually compiled they actually devised the different, all the different procedures, starting from sample preparation, then RNA isolation, cDNA synthesis, and finally to real-time PCR and further analysis, how it should be done in a perfect manner. So that was that was devised, and they published an article, and it is known as MIQE guidelines, that is minimum information for publication of quantitative real-time PCR experiments. So this paper explains what all things, what all checks and what all uh, uh, I mean, uh, what all analysis you have to do when you do a real-time PCR, so that the result that you got will be accurate. It will be perfect. So they have, and also when you publish real-time PCR data, what all results you have to show along with your actual result, what all other information also you have to provide for the journals, so that they can verify the result is accurate. So anyone who does real-time PCR or are planning to real-time PCR should read this article, and that explains how to do the real-time PCR in a perfect manner. And also they standardize the way in which different terminologies that we use in PCR. Those were also standardized. Like earlier, people used to refer real-time PCR as RT-qPCR and QRT-PCR, etc. So there were a lot of confusions, so they standardize all those things also. Like now Qpcr refers to quantitative real-time PCR. So Qpcr means quantitative real-time PCR using DNA, that is Qpcr. And if you are using RNA, then that becomes RT-qPCR. That is reverse transcription quantitative PCR. So if you are using real-time PCR and your template is RNA, then it will be RT-qPCR, that is reverse transcription Qpcr. So these are the standard terminologies, and I will discuss some of the important aspects of MIQE guidelines for Qpcr. The quantity and quality of mRNA is very important. So before you do real-time PCR, you should check the quantity, accurate quantity of your mRNA, and also the quality of mRNA should be ascertained. So you can do a A260, 280 analysis to identify the quality of your mRNA, and also it's important to calculate the integrity of your RNA. That is, your RNA is not degraded. That should also be ascertained by some assays like RIN analysis, that is RNA integrity analysis, or by comparing the 5' and 3' of your RNA by designing two different amplicons to your 5' and 3' ends, and then doing a real-time PCR. You can identify whether your RNA is degraded, or your mRNA, the gene that you are studying, its mRNA is degraded or not. So the stability of the mRNA should be confirmed. That's one aspect. Then another important problem is DNA contamination. When you isolate RNA, you will have DNA contamination also. So DNA also will be there, and so you should ensure that the DNA should be completely removed, because when you do real-time PCR for gene expression, uh, uh, your amplification will be coming from the genomic DNA. If you have genomic DNA contamination in your sample, the amplicon will be coming from the genomic DNA also, because the primer can bind to the genomic DNA also. So so that will affect your real-time PCR result. So that should be taken care. So DNA should be completely removed. And also another thing you can do is you can design your primer, any one of your primer, to an exon intron junction. So if you design a primer spanning the exon junction, and then that primer will not bind to the DNA template. It will not bind to DNA, it will be binding only to your cDNA coming from the mRNA. So that's another way of avoiding amplicons from your genomic DNA. Then when you do cDNA synthesis, you have to take equal amount of RNA. That's very important. Then when you design primer, what all things you have to take care. That also is that also is dealt in detail in the paper. I'll not go into the details. Okay. Then the Qpc optimization is also important. When you do Qpcr, you have to do many other analysis also, so that you can ensure your Qpcr is perfect. So one thing is primer specificity. That is the primer that you are using. If you are using Cybern chemistry, this is very important. The primer specificity should be ensured. That is, there is no primer dimer, and also the primer is not binding to unintended targets. That's very important. Another important thing is the efficiency of your primer. When we did the relative quantification by delta CQ method, we took the amplification efficiency as maximum. That is, in each cycle, the PCR products doubles. That's why we took 2 in 2 raised to minus delta delta CQ. That 2 stands for, that's the amplification factor. So if your amplification efficiency of your PCR is maximum, that is 100%, then the amplification factor is 2. That means every cycle the PCR product is doubling. But it need not be so, because if your primer is not efficient, very efficient, it is having only 98% efficiency instead of 100, then that amplification factor will not be 2, instead it will be 1.98. So that will affect your calculation. So the relative quantity that you are calculating will be affected if the primer efficiency is not 100%, as you infer when you use 2 raised to minus delta delta CQ. So there are softwares where you can calculate this primer efficiency and then put the actual value based on your efficiency. So if your efficiency is 98%, instead of 2, actually you have to put 1.98 raised to minus delta delta CQ. Then only the fold change will be exactly correct. So efficiency of the primer should be confirmed, and for that, either you can do a standard curve analysis, wherein you will be making a dilution of your template, and then yeah, like this, you'll make a dilution of your template, and then you'll do a you'll generate CQ values for the dilutions, and then using that data you can create a standard curve, and from the slope of this curve you can calculate the efficiency of the amplification. So basically whether the amplification is happening at 100% efficiency or less than that, that can be estimated. Even the real-time, the software that come with the many real-time machines, in that you can do such an analysis and you can estimate the efficiency of your primer. And also, and there are also simple softwares which tells you the efficiency of PCR from the amplification curve itself, like the Linreg software. This is a freely available software, and using this software from the amplification plot itself, that is this amplification plot itself, from the data points you can identify whether the increase is happening in an exponential manner. That is, whether the increase is happening doubling in each PCR cycle, the PCR product or the fluorescence is doubling. So there are softwares which will analyze the amplification plot and tells you the efficiency of the PCR. So in a perfect manner, if the real-time PCR is to be done in a perfect manner, you have to actually estimate the amplification efficiency of your primer, and then using that you have to calculate the relative values. And then there should be negative controls like non-template controls, where you don't add template, and there should be no reverse transcriptase control, where when you are using mRNA, you should convert the mRNA into cDNA using reverse transcriptase enzyme. So there should be no reverse transcriptase control as a control for that reaction. And there should be technical replicates and also biological replicates for your samples. So these are some of the points that you have to keep in mind when you do a real-time PCR. And these are also said in the MIQE guidelines. And another important thing, as I mentioned earlier also, the selection of reference gene, that's also very important. If your reference gene is not stable, the reference gene in the earlier case, we have used actin. So normally people use actin, and they blindly take actin as a reference gene for all different assays. But if the gene that you're taking, like actin, is not stable in your particular experiment, if it is affected by your treatment, then the data that you are getting will not be correct. So that's the most important aspect in a real-time PCR assay. When you do relative quantification, you should ensure that the reference gene that you are taking is stable in the samples that you're studying. It should not be affected by your treatment. So there are no universal reference genes. You cannot blindly take any reference gene like actin as a reference gene for any experiment. So when you select your reference gene, you can search literature whether there are prior works similar to your work. If there are prior works, and if they have they have identified some stable reference gene, then you can blindly take those reference genes. If such data is not available, then you have to actually do an analysis with different possible reference genes, different possible reference genes, and then you have to do a analysis to identify which one is stable in your sample. So there are many softwares available for doing such analysis, like Genome, NormFinder, BestKeeper. Genome is a priced software now, but NormFinder and BestKeeper are uh, open source software. They are simple softwares based on Excel, so they are available. So there are many softwares like this, wherein you have to identify some uh, genes which can be stable in your study, like some housekeeping genes you can select, and then uh, you have to generate CQ values for these genes in your different samples, and then that data you can import into any of these softwares, and then using different algorithms they will tell you whether these genes are showing or which gene among these is the least varying one, which is the most stable one, in other words, among your sample. So they use slightly different algorithms. So instead of going for one algorithm, you can go for many different algorithms, and then you can decide which gene is stable. So this is also possible. So if there are no prior information about stable reference genes in your particular kind of experiments, then it's always better to identify a stable reference gene first, and then go for the real-time PCR. Okay, this is how the genome analysis looks. So they have used many different genes, and then after analysis it will give you a value, the genes with the lowest value, that will be having low variation. So like this, you have to take different genes and then generate CQ values using your samples, and then do any of these analysis, and that will tell you which gene is stable in your samples. Okay, so I think with that I have completed some of the basic aspects of real-time PCR and the applications of real-time PCR. And if you have any doubts, and if it's possible, I can answer.
Thank you so much, sir, for sharing your knowledge on the topic real time PCR, applications, and significance. It was really informative and descriptive. Thank you so much, sir, once again. Now, let me invite Dr. Santhya Vincent, Assistant Professor of Research and Postgraduate Department of Botany of St. Thomas College, to deliver the vote of thanks. Welcome, ma'am.
Thank you, Helena. Good evening, all. Today, we have come to the last session of our lecture series on recent trends in biotechnology. In this session, we had Dr. Kiran A, Assistant Professor from Kerala Agriculture University, who gave us a lecture on real-time PCR, its applications and significance. This lecture series was aimed at creating awareness among young aspirants in research fields about the recent and advanced trends and techniques in biotechnology. It was a very fruitful session as a resource person very well explained about the procedure, significance of real-time PCR, along with the data analysis, as well as the way of publishing articles using this data. The talk given was based on his experiments and experience, which made this session much more interesting. He also explained about the science behind the PCR technology and its applications, and also hinted on the several doubts which one will have when using this technology. So, on behalf of the entire botany department of St. Thomas College, I extend our gratitude to Dr. Kiran for handling this session so well. Thank you, sir, for this wonderful session. I also thank all the participants
Thank you,
for
ma'am.
being part of. Yes, yes. Thank you. Thank you, sir, for this wonderful session. I also thank all the participants for being part of this lecture series. Hope it was very effective in increasing your knowledge level. I also thank all the organizers, especially Dr. Geetu Elizabeth Thomas, for conducting such a wonderful lecture series. Once again, thank you all. Thank you.
Thank you, ma'am. Now, I request all the participants to kindly fill the feedback form in the comment section. And with this, we wind up the session. Thank you.